All
right, everyone. What was that?
Oh, nothing. It just gave an announcement. We started the recording. We're good.
Great. All right. Thank you, everyone, for coming. This is a welcome treat to actually have an in-person presentation for a change. Glad to see you all here, appropriately socially distant. They're giving a shout out to Kavya. And thank you, Kavya Charlin, who is our teaching assistant for the class portion of the seminar series and who has helped organize a lot of the seminar series herself. But we're very fortunate today to welcome one of our very own Vanderbilt faculty members, Dr. Shane Hutson. Just a brief background into his academic history. He got his Bachelor's of Arts in Physics with a minor in Mathematics and a Master's in Physics from Wake Forest University. Went on to get a PhD in Biophysics from University of Virginia and did a postdoc in Biological Physics at Duke University before joining the faculty here at Vanderbilt in 2003. He has worked his way up the ranks and is now a full professor of physics and biological sciences and currently serving as the chair of the Department of Physics and Astronomy. He's also the deputy director of VBRE, that's how I pronounce it anyway, Vanderbilt Institute for Integrative Biosystems Research and Education. And he's the director of VPrompt, the Vanderbilt Pittsburgh resource for organotypic models for predictive toxicology. His research has been focused on understanding the process of tissue morphogenesis, and his work leverages the powers of mathematical and computational modeling. We're really looking forward to hearing his talk today. It seems like most of it will be on a recent publication that came out in Developmental Cell in collaboration with Andrea Page McCaw's lab, investigating calcium signaling propagation in epithelial wound sensing and repair. So thank you.
Thanks, Aaron. Let's see if we can get this set up well here. Oh, you know what? We're not projecting it up here. That could be a problem for everybody who's actually live.
Now yeah, that's right. The live people won't be able to see that. So this goes here. This should be.
Yeah.
No, this one. It's gone. All right, we're good.
All right, good. We got to swap displays here.
Try presenting it.
There we go.
Okay.
And then
All
you
right.
can share your screen on Zoom.
Yep.
Good to go.
Let's see. To start sharing before I do the full screen there.
There you go. I'll get rid of that. It's just.
Right there.
I can't make that go away. Let's just move it out of the way.
Yeah, just move it. There we go. And then we can close that one. Maybe that'll get rid of the other two. Okay.
All right.
All right. Sorry about that. Since I'm distance from everybody, I'm going to take the mask off so I can talk and be up here.
One quick note, real quick.
Yeah.
Sorry, Dr. Hudson.
Nope.
Could you swap the displays on the Zoom? Right now, I'm seeing presenter mode.
So the
There's-
difficulty is
Oh,
that
I know what's happening. Yeah.
we have it on both of them. I have to mirror instead of doing that. Let's see.
So, yeah, do
I got to mirror the displays instead of. It is set up to mirror them.
yeah, I think this is a Zoom thing. You'll have to deal with it.
Yeah, I'm sorry. I don't see a way to get rid of it. It's one of the presenter view is going to show up either for the people online or the people in the room.
Got it. It's fine. Yeah,
Okay.
all good. This is perfect.
All right. So thanks for bearing with us. So I will say from the start, right? Darren mentioned this. This is collaborative work with Andrea Page McCaw's lab. She's in cell and developmental biology, and I love this kind of research because it's stuff that my lab wouldn't be able to do on our own, and her lab wouldn't be able to do on its own. But we can combine our talents and do much more than either one of us could do by ourselves. So this is cell biologists, physicists, mathematicians, all working together to solve the problem, right, to tackle it. Now, I did go above and beyond for you all today, and I'm not talking about the tie. This is completely irrelevant. Something else. I went for a run on Monday afternoon, like I do most afternoons around here, and what must have been just an incredible act of carelessness, I ended up kicking a rock on the sidewalk and tripping myself, face down. Right, managed to save my face from hitting the sidewalk, but my hands got torn up, and so, I'm not going to show you the wounds. I'll keep those covered, but this is all about wound healing. And normally we think of that as a pretty routine process, right? I got to brush myself off and I'm like, well, in a week or two, those will be healed up. It's not that big a deal. Most of the time that's true. But there are examples where wound healing doesn't work right? In particular, mostly for diabetic patients, it can be very slow or can not happen at all correctly. And on the other side, there are examples where chronic wound healing should shut off and it doesn't appropriately. Right, that's thought to play a role in psoriasis. It's part of what leads to atherosclerosis in your arteries, little wounds to the interior of the arterial lining over time. And in many ways, it's what happens in epithelial-based cancers. Right, you get this sort of proliferative response to respond to something that's gone wrong in the local environment of that epithelium, and it just doesn't turn on. Right, and so when we start looking at this, although it's routine, there's some really fundamental things that we just don't know yet, that we don't understand completely. And I'm going to tell you one of those stories today about something that we've just learned and just figured out about wound healing, and that's how the cells know that there's a wound there in the first place, right? How they detect its presence. So make sure this will all work correctly. Okay. So first step towards doing that is to recognize that we're going to be looking at epithelia, so just sheets of cells. And cells in an epithelium around a wound respond in a patterned way. It's not just the cells directly adjacent to the wound. When we go out multiple cell rows, the cells all respond, there's a large number of them, and they do different things depending on how far away from the wound they are. The ones closest in are actually mostly migratory and sort of fill in as quickly as they can. And then when you get further away, those are the cells that actually proliferate and add more cells to the mix to help fill the wound up. Now, we can look specifically at our system, and in ours, we're making really small wounds. We're using a laser to do it. So these are wounds that are 10 to 50 microns in diameter. And they heal up over the course of about 6 hours. So we can really follow the whole process and see what's going on with them. That was a labeling of the nuclei to see how that happened. Interestingly enough, we can even just looking at the nuclei start to see some patterning. We can use a Fuji stain. I don't know if you're familiar with that or not, but it sort of indicates where cells are in the cell cycle. They show up different colors depending on what part of the cell cycle they're under. And you notice the yellow right around the wound, those are cells that are stuck in sort of this G1S transition that can't seem to progress. Those are the cells that don't divide in this case. And if we look in a little bit closer and stain the nuclei for how much DNA is in them using DAPI, we get different amounts of DNA in cells close to the wound than those further away. Right? So there's very interesting things going on with cell replication around the wound. And there's also this fascinating response where, see these large cells like this? That's actually the fusion of something like 10 to 15 cells that were around the wound. They lose their cell borders, and so we have one big cell membrane around 10 to 15 nuclei there. And that seems to be an important part of the wound healing process. And that only happens for the cells that were really close in around the wound. As we move further back, we don't have that. So we see this evidence of patterning. And I will say that sort of following up on what's going on with the replication, what's going on with the cell fusion, that's the future directions of our research. I'm going to tell you sort of a story behind that and what we go to next. And so that story, right slide, is a story of calcium signaling and wound detection. So one of the most ubiquitous and earliest signals we see around wounds in all kinds of organisms, right? C. elegans, zebrafish, fruit fly, mouse. You make wounds either by puncture or laser or snipping with scissors. And one of the things you see is that calcium rushes into the cytoplasm of the cells around the wound, right? It's ubiquitous second messenger, that's not surprising, but it's one that's used extensively in wound repair. And so now we're going to make our wounds on the back of fruit fly pupae. So this is a nice part of the fruit fly life cycle because they're immobile, and yet there's fully developed tissues in there. So we can take it, it's like the cocoon of a butterfly, but it's for a fly. Open up that pupal case, and it exposes this beautiful epithelium on the back, or the so-called notum of the fly, and we can do our experiments there. And now here's Erica Shannon, who was a grad student in cell and developmental biology, who really got us all started on this project. She came over to my lab, wasn't afraid to play with the optics, jump in there, figure out how to make the wounds with the laser. And so we're going to bring in this external laser, make very small and reproducible and controllable wounds in the pupal notum, and then track what happens around it and use that to figure out what's going on in terms of wound response and wound detection. So this movie played at the start when we were fiddling with things, and so it probably got overlooked. This is the dynamics of calcium signaling around one of these laser-induced wounds. Right? So the cells here are expressing GCamp. It's a fluorescent indicator, right? It's a GFP variant. When it binds calcium, it folds up appropriately and fluoresces. And you can see that, if I go back and play this again, there's sort of this initial burst. There's a lot of calcium there. And then it later goes into the second expansion into further regions away. And then we have these dynamic flare or flame-like propagation of the calcium signals around it. Right? Now, one of the first papers we did on this a couple of years ago that Erica really led, and I'll show you some of it, was sort of, as a physicist, what I would think of as the kinematics. We just described what do we see, right? What do the signals do as a function of time, and how do we relate them, and what's consistent, what's not consistent. And now we're going back in and looking at what's the mechanism, what's driving it. Because this is the first signal. This is like the cells, the cells raising their hand going, hey, something's going on here, right? But something happened to happen upstream before that. And we want to figure out what happened upstream before that, right? How is it the cells know what's going on at this point? You will also notice that sort of this dynamic property gives the cells a chance to figure out not only that there's a wound nearby, but how far away from it they are, right? If they look at just what fraction of the time or how often does calcium spike up where I am, they can use that information to figure out distance from the wound. All right, now let's take this from a single cell perspective and think about it. When we look at this, think about the spatial and the temporal patterns, but if you're an individual cell in the epithelium, all you know is what's happening locally, right? And so for some of the cells near the boundary of the wound, right near the margin, calcium goes high and essentially stays high for a long time. Some that are a little bit further out, there's an initial burst of a couple of minutes where calcium stays high, and then it drops back down. You just get these periodic spikes when one of those flares happens to cross over where that cell is. And if you're far enough out, you don't get that initial burst of calcium, but you might see a couple of flares pass by. So you get a couple of calcium spikes here, right? And it's this kind of information that the cells could be using to figure out where they are. Now, let's think a little bit about what could be upstream, right? If we're sitting here in the room and you all are cells in an epithelium, so try to put yourself in that position, right? Lightning bolt strikes, Zeus throws a lightning bolt out here, and Darren is vaporized. Sorry. Okay. You all would know about it instantly because you're using your senses, right? You see it, you hear it, maybe you smell the acrid smoke left over, right? You would pick up all these signals. But now, put yourself in the position of a cell. A cell doesn't have all those same sensory inputs. Right? What sensory inputs does a cell have? Certainly you're blind, right? You can't see. Pressure, right? Pressure, mechanics, anything like that. So that's a possibility, right? When one cell in the tissue is wounded, it loses its connections to its neighbors. So you could have both pressure associated with making the wound, right? It can create shear stresses in the vicinity, and changes in tension associated with the cells being missing and things relaxing around there. Yeah, so that's absolutely one. What else is possible in there? There's possibility of electric fields changing, voltages changing, those kinds of things. Yep, that could be part of the process. What other senses do cells have? The individual epithelial cells have?
Signaling molecules through
Yep.
and surface line.
Yeah, there's signaling molecules, right? In essence, it's the analog of our sense of taste or smell, right? They can chemically communicate with one another. And so we've got to look at all those possibilities, changes in the mechanical aspects, changes in electrical aspects, changes in what signals are released where, what small molecules might be floating around. And all of those have been hypothesized. And in certain instances in other wounding systems, one or the others of them have been found, right? This particular system, no one had investigated yet and figured out what was going on with it. So as we get ready to go through this, let me give you a brief overview of the kinematics as I described it. So we have some language to talk about this. So we make the wound, and the earliest image we could take at this time was 2 seconds after the wound. And already there's calcium in the cells that goes out about 50 microns for this size wound. That initial influx of calcium then spreads out in what we call the first or localized expansion. It goes out 1 or 2 cell diameters. And here, where we're tracking the radius of the calcium signal as a function of time, that's the little red part of the curve here where it goes up in these 4 different examples. The calcium expansion then pauses for something like 40 to 80 seconds. It varies between different wounds, and it even retracts a little bit. That's where the black line goes down here. But then there's this second expansion that's in the blue that goes out to much more distal cells and recruit them, and they then know about the presence of the wound. Right? And so we'll talk about this first and second expansion, or local and distal expansion, and trying to figure out what's behind that and what the different mechanisms are for those processes. So the overview from here, we'll talk about what drives that initial short range expansion, that first one, what drives the latter distal expansion, that second one, and then highlight what computational models can do to confirm the working hypotheses, to help us make new predictions, and to test those predictions. All right, so what's going on with the first one? This is the shorter of the stories, and it's mainly a physics story, actually, in the end of it. So I told you when we were taking the images, we saw that initial calcium influx in about 2 seconds. The microscope system we were using at the time was limited in that regard, but it was a laser scanning confocal microscope. So we could, instead of trying to take a whole image, just scan one line. It's like our image is one line. We can make chimegraphs out of that. And if we do that, then we can see, we make the wound right there. Time is going down on this axis. We see calcium rushing into these cells 50 microns out in less than 10 milliseconds. So this is an extremely fast process. It's much too fast for any sort of diffusion of this small molecule that could take place. We thought maybe it was electrical, maybe it was mechanical. We weren't really sure at that point. We started investigating what could be driving these different parts of it. And turns out that the way we were making the wounds actually allowed us to figure this out. Right, so we're making them with a laser. When you make a wound with a laser, what's happening? Well, at the focus of the laser, the first thing you're doing, it's a pulse laser. It's very high intensity, very strong electric fields. It's actually ripping electrons off of the water molecules and the macromolecules and creating a plasma. Right? Those electrons then recombine, often not with the place they came from, and that can cause all kinds of chemical effects and chop everything up. But it also leaves that area very hot. Right? It puts a lot of thermal energy in there. Biological systems are completely in water, right? It's an aqueous environment. If you get a really hot spot, that vaporizes it into water vapor. That water vapor expands as a bubble. And actually, it turns out there's some very nice physics that goes back to Lord Rayleigh. That bubble expands out to a certain maximum radius and then collapses again. Right? And it can, in a very symmetric environment, bounce a few times. But let me show you sort of what this looks like. If this is the epithelium seen from the side, this is from the top, and that bubble will expand out and collapse again. And so there's some footprint of it. Now, the problem is that bubble expands out and collapses in a matter of microseconds. So much too fast for us to pick up with the microscope as it's normally configured. Right? We can set it up so we do some flash imaging, where we basically have an ablation pulse, have a imaging pulse that arrives a few nanoseconds later and pick this up, and you can just see some like little shadows like this of these cavitation bubbles. Very difficult experiments, but possible to do. The easier way to do it is just to put a microphone over the sample and pick up the acoustic transients. When that bubble expands, you get this big shock wave, and when it collapses again, you get another shock wave. You have a question?
No.
Okay.
Impressive.
All right. So you get this pair of shock waves, and you can use the time between them. They're like, okay, the bubble lasted 5.8 microseconds. Go back to Lord Rayleigh again. Lord Rayleigh worked out all the physics of these bubbles, and if it lasted 5.8 microseconds, we calculate exactly how big it got. Right? And so we know it got to, it's about a multiple of 5. So it's about 30 microns for its radius there. We worked that one out. So. And if we do this in an embryo, we don't get quite as clean a signal. This was in a nice, just plain water, but you can pick up those 2 shock waves. And we can track it. Yes?
Have you noticed the same response in the time for different movies? Like you get a factor 5?
Oh, for different animals? I don't think anyone's ever looked at that, right? So the factor of 5, right, what comes into that? It's the density of the fluid in the surroundings and the ambient pressure both play big roles. Viscosity plays a little bit of a role. And so you would expect it to vary, but those 3 parameters shouldn't vary that much from one organism to another. All right, so we have a way, yeah.
So there's a cascade of new feeling that is in humans, as in the calcium is involved in that, the physiology and all of that. So does that happen in Drosophila as well in nowadays as well as happening? I mean, because the calcium is really important in humans for the living. So I'm just trying to compare it with things.
So the calcium is important.
Dr. Hudson,
Yes.
sorry to interrupt. Would you mind repeating the question just so we can also hear?
Certainly. So the question was, to what extent do we know that calcium's important for the wound healing process in fruit flies, right? We know it's important in humans and other organisms. How important is it in fruit flies? So one of the difficulties with answering that is there's not a way to knock out all the calcium signaling, right? There's calcium ubiquitously in the fruit fly pupae. We can't get rid of the calcium. All we can get rid of is sort of the mechanisms that allow it into the cell. And I'll show you once we get through this a little bit that we can knock out some of those mechanisms, and we do see a decrease in survival after wounding. So it is important. Yeah. Okay, good question. All right, so we got the cavitation bubble, and then we can plot the maximum radius of that cavitation bubble for any individual wound, sort of the footprint of it, and compare that to the radius of the initial calcium signal. And they match up 1-to-1. Right, so it is the footprint of the cavitation bubble that's allowing that first bit of calcium to come in. Okay. And now I'm not going to show you all the other results, but we looked at depolarization of the cells, we looked at ability of dyes to get into the cells, and they all go in the same footprint. Right? And so what it suggests is what the cavitation bubble has done is basically collateral damage. It's popped open micropores or little micro tears in the plasma membranes and allowed calcium just to come into the cell, right? Extracellular calcium is something like a millimolar, intracellular calcium is a few micromolar. And so it just goes rushing in when there's a tear there. Right? And so it's very much a physical process that gets the first calcium into the cells. And there's no way for us to knock that out. Although we figured out some stuff that I can talk about in question and answer, where we might be able to do that. All right, so this is very touchy. Okay, the first part of the story, right? It's a physical influx of calcium due to just collateral damage. But now it does expand out of cellularity, right? And to understand that, then we had to start combining with the biologists to figure out some genetic tools to tackle this. Now, this was beautiful work done by Andrea and graduate students James O'Connor in her lab, where they could actually take the nodum of the fruit fly, and although it's a single continuous epithelium, it's got different genetic domains in it. And so we can use the fruit fly genetics to manipulate one group of cells and not the other group of cells. And in this case, we're manipulating the cells that are labeled in red here. That's called the pannier domain, which is sort of down the middle of the back of the fruit fly. Right? And we're going to make the wounds then on the border between the pannier domain and the normal control domain that's adjacent to it. And then we can have this internal control and say, we make a wound, do we get symmetric responses on the two sides, or do we see something different on the pannier and the control side? Right? If we do this and we really just labeled it, right, didn't manipulate the genetics in any other way, we get the same response on both sides when we make a wound. On the other hand, if we do something, for example, as a start, stop that one for a second. We're going to make a wound on the border. In this case, we're making the wound on the left border of the pannier domain. So this is the region where we're going to manipulate stuff. And what we're going to do over here is knock down a protein called annexin 2, which is the gap junction protein in Drosophila. Right? So gap junctions are the connections between epithelial cells that let small molecules flow from one to the other. We're going to eliminate that on one side, and gap junctions are going to be fine on the other side. Okay? And so if we do that, and then make the wound, there's a couple of different things going on here. And so for our story thus far, I want to focus on the first of them. Stop this at the right. Notice that calcium came in and sort of stayed in that configuration for the first part. But over here on this side, it expanded out of cellular too, right? And so that was calcium going through the gap junctions. So there's collateral damage that lets calcium directly into the cells. It can go through gap junctions to one or two cells further out. And gap junctions are also playing another role. Notice on the right-hand side here where they're knocked down, that we get like this speckled appearance. Cells are behaving very independently from one another. Right? They help coordinate the behavior when they're present, and when they're lost, the cells behave very independently. All right. Now, that's story one. Story two, trying to figure out what's going on with the distal calcium expansion, right? That later delayed expansion of what's going on. So we had some hints that it would be related to IP3 receptor and the GQ pathway, because that's a very common way to get calcium release in cells. So we went through and we knocked down different parts of that. We'll knock down either the IP3 receptor itself, which lets calcium out of the endoplasmic reticulum, or we'll knock down PLC beta, the enzyme that makes IP3 that binds to this receptor and lets calcium out, or we'll knock down G alpha Q, a little G protein that actually activates PLC beta. And so we have these three different steps in the pathway we can knock down. And if we do that, in this case, the knockdown side is the left, And you'll notice that it's going to behave differently from the control region on the right in each of these cases. And we get that nice second or distal expansion on the right side, and we don't see it on the left side. We can quantify it down here in terms of the radius of both of those. All right. Questions so far? Okay. So this is where we were when we put in our grant proposal, and we said, oh, it looks like it's a G-protein-coupled receptor. Here's what we're going to do. There are only 100 G-protein-coupled receptors in Drosophila. So we are just going to brute force go through and try them and figure out what the receptor is here. And the reviewer said, yeah, that's doable, probably. It's a lot of effort. And we're worried that in the end, it might not be a G-protein-coupled receptor, but might be some sort of receptor crosstalk. Right? Maybe really there's some receptor tyrosine kinase. There's several of those known to be involved in wound responses, and that it's somehow working through the G pathway, right? Or it's coupling into the G protein pathway. So we were worried. We were preparing our next submission, and then we got word that it was funded. So they let us go ahead and do it anyways, despite the reservations. And so when we got ready to do this, to the credit of a very clever graduate student, James O'Connor, he sat down and made this huge table of in what tissues are all these G protein coupled receptors expressed, which ones couple the GQ pathway to the GI pathway to the GS pathway, and which one have ever had any papers that seem anything to do with wound healing. And he made a prioritized list out of it. And lo and behold, the third one he tried turned out to be the receptor, right? And so the receptor that's involved is something called Methuselah-like 10. It gets that name because if it's knocked down, the flies actually live longer. So it has some relationship to lifespan in the flies. So we're going to knock it down on the left side, control side on the right. And if we do that, we get a nice second expansion on the right side and not on the left side. We tried several other candidates that we thought would be important, like the adenosine receptor and some of the receptor tyrosine kinases, and none of them gave this similar response. All right, so we got the receptor. Now we got to figure out what the ligand is, right? We're sort of working our way back through this process. Now, to your question earlier, right? So how important is this? We can knock down this receptor now that we have something that's very specific for the calcium coming in around wounds. And if we do that and we make wounds in the nodum, most of the time in control organisms, almost all of them survive. It's like 95% survival rate. We can drop that down to about 70% if that pathway is knocked out. Right? And maybe if we made larger wounds, that would be a larger fraction, right? We would have less of them surviving. But it's certainly, there's an undeniably significant effect of knocking that down. All right. So one of the other things that we wanted to tackle is, and started looking at the ligand, is this a ligand that's released by the wounded cells and then diffuses in the extracellular medium? Or is it something a little bit more complicated where they're signaling from one cell to its neighbor and then from that cell to its neighbor, some sort of bucket brigade mechanism that propagates the signal outwards? And so we can do this experiment here where we're going to actually make the wound completely in the pannier region where we knock down G alpha Q, so we shouldn't get a second expansion over there. But we're going to make it close enough that maybe if there's a diffusible signal, it could reach over here and trigger an expansion on that side or trigger a calcium signal over there. And that's exactly what happens. We move that over. Wait a second, nothing's happening. And then it shows up on the far side over there in that tissue, right? So we've got something extracellular diffusible that doesn't require this bucket brigade mechanism. That's then the ligand for the Methuselah-like 10 receptor. Yeah.
So, I mean, at the cellular scale, diffusion of something from that point to that far distant point seems a little impossible. I mean, how is it happening? Like simple diffusion wouldn't be fast enough to transfer that, right?
Well, actually, it is in this case, right? It's a matter of scales. We're working on sort of 100 micron length scale, and we're talking about something that takes place over hundreds of seconds. And actually, on that length and time scale, diffusion is perfectly adequate. We can actually, based on how quickly the signal spreads, calculate an effective diffusion constant. And it implies a really large protein complex. Right? It doesn't have to be a small molecule that diffuses really fast. Yeah. So diffusion can work, at least on a theoretical basis. It's enough. Yeah. All right, so there was existing literature on Methuselah-like 10. It was known to bind to these peptides called growth blocking peptides. There are 5 of them in the fly, but 2 of them are expressed in the adult in all tissues. The others are expressed in specific places at different times. Now, these peptides are sort of produced ubiquitously. Right? They aren't local. Like the ones in the pupil nodum aren't made by cells in the pupil nodum. They're actually made by the fly equivalent of the liver and then sort of circulate throughout the fly. So we can't do the same sort of split system experiment that we did before. But we can take an example where both of those growth blocking peptides 1 and 2 are knocked down, and we make the wounds in those organisms. And if we do that, we see that first expansion, but we never get a second expansion. And then if we want to figure this out a little bit more, we have to go to a slightly different experimental system. In this case, for those of you who are familiar with the fly, as the fly is crawling around as a larva, there's these little circles called imaginal discs, little discs of cells, and they will become the adult tissues during metamorphosis, right? The cells that will become the nodum are the wing disc, and so we can actually extract wing discs from fly larvae and do the experiments on them. And so we took the wing discs out, and we can take them from different genetic backgrounds, and then we can just add whatever chemical we want onto the top of them and see how they respond. So we made some synthetic GBP1, and if we add that onto wing discs, we're going to add it onto some wing discs that are control or wild type, and some that have Methuselah-like 10, the receptor knocked down. And if we do that, then we get a calcium response in control, but not when we knock the receptor down, right? That's the signal we want to see. That's a good sign. We can do it with GBP2. It's just a different dose response relationship for the 2 of them. It gets a little bit more complicated because, all right, we do know that these ligands now can signal through this receptor. We don't know for sure that that's what's going on in wound healing. We know that if we knock those ligands down, we don't see the signal in wound healing. Right? There's a few unresolved questions around the edges of this. There's also a long-standing observation that you can take what's called fly extract, which is basically just ground up fly or larva, and put it on these wing discs, and they will light up with calcium. Right? Calcium comes rushing into them. Right? That mimics a lot of a wound, right? You take a lysate of the cells in a fly and you put it onto other cells, and they recognize something in that. Right? And so here we can take fly extract or larval extract and put it on these wing discs and show that those light up or not in that same receptor dependent manner again, right? So whatever fly lysate does, it's also acting through this particular receptor. Right. Now, as this may be a case where we pushed one experiment too far, but we learned something really interesting after being confused for a while. So we're going to take extract that came from flies that either did or did not make GBPs, did or did not make the ligand. And we're going to put it onto wing discs that either do or do not make the ligand. Right? Now, if we have GBP in the extract, but not in the wing discs, the delta. We're adding extract that does not have GBP. Sorry, I said that incorrectly. So if we take extract that doesn't have the growth blocking peptides, we add it to wing discs that don't have the growth blocking peptides, we don't see anything. That makes sense. But now the other combinations. Okay. You'll notice that 3 of the 4 up there lit up. So it makes complete sense that adding control extract onto control discs lights up. But these other 2 are a little bit odd, right? So control extract on the discs that don't have GBP, that makes sense. We expect the GBP to be in the extract and the receptor to be on the discs, okay. But this one lights up too. We take extract that doesn't have GBP in it, and we put it on the wing discs, and they light up. So where's the signal coming from in that case? Took us a while. You want to answer?
Well, didn't you say it was like ubiquitous throughout the system, so it was already on?
It is already on the control tissue, right? That's a big hint, right? It's already in there because it's ubiquitously expressed. So why is it the control tissue lighting up all the time? Why is it that adding the extract all of a sudden causes it to light up? And there's an important part of processing of those growth blocking peptides that becomes important. They are actually secreted as this propeptide form, and they're sitting around the extracellular matrix in this propeptide form, and they require a protease to cleave them to activate them. And so now the story's gotten more interesting. Right? It's not that just growth blocking peptides are released from the cells, but they are activated by something that's released from the cells. Right? They were already present. And they were already present on the wing disc, and we added the extract, it activated them. Right? Now, the candidate for activating them is obviously proteases. You lyse open a cell, you're going to release a lot of proteases. Right? And so we went through and tested this. We took trypsin as just one example, or clostrapane as another class of proteases. And if we added either one of those onto wing discs, the ones where the wing disc itself had the GBPs lit up, the ones where the wing disc didn't have the GBPs didn't light up. And so putting all of that together, we have this model where, all right, we make a wound, and one of the things that happens is proteases are released from that wound. Those proteases cleave the propeptides, the pro-GBPs that are out there in the extracellular matrix, releasing a fragment of it that can diffuse and bind to receptors and activate the calcium signal. So that's the working hypothesis, what we've been able to get to with the experiments. And that's the end of story 2. Right? So story 3 is now, should be interesting to you all as systems biologists. How do we put this together and model it and think about what that tells us? So there's 2 levels of models I'll tell you about quickly here. One is just trying to understand what's happening extracellularly. So we can put this in. This is not a terribly difficult system to model. We're going to have some protease released from the wound in some spatial and temporal manner. It'll be able to diffuse, bind to the pro-ligand, cleave it, release the ligand, which can also diffuse, and that ligand can bind to a receptor reversibly, and then we'll trigger calcium signals from there. Now, we can track any of the different parts of this. For example, here's the protease being released from the wound. It actually turns out to match the experiments. We have to have the protease released fairly slowly. It doesn't get released instantaneously. We can see over here that the blue curve is the ligand itself building up. The golden curve here is the pro-ligand being depleted. Here's the amount of receptor, or the fraction of receptor bound to ligand or not bound. And what you will be able to see, let me reverse this and start it over, is that over time, the bound receptor goes up. And if we track some front on it, we track just a threshold, like where does it cross 50% bound? As just an example. That moves out away from the wound with time, and we can track that as a calcium signal radius versus time. Admittedly, arbitrary 50% is an arbitrary threshold, but you can do this with any threshold that you put in for it. Okay. And if we do that, it'll fit really well to the curves for these second expansions. Describe them, right? We can't match up what's happening here. This is a different process that's not in our model, right? And it's sort of hitting what's happening here. But this works really well for describing it. It does take a little bit getting used to as a model, because it's in this class of models that are called soft or fuzzy. Old school, I'm very used to, I take something and fit it to data, and I get really nice estimates of what all the parameters are in there. This kind of model does not do that. This kind of model, we get sort of order of magnitude estimates for the parameters because there's coupling between them. Right? And we say, well, we could increase this one, but it would, or decreasing this one would do the exact same thing. Right? But we can look at this and see what the effects of varying all the parameters, there's 9 parameters that are actually in here, what all of them would do. Some of them have almost no effect on the calcium radius versus time. Others change sort of the delay time, others change the speed with which it moves out. We can look at all of those. Importantly, we don't really have a knob where we can change any of those parameters in the experiments to test it. Not directly. What we can do is there's two of these. Sorry. The size of the protease source and the mean protease concentration that depend or should depend on exactly how big a wound we make. Okay. And so if we mimic making wounds of different sizes, those wounds would impact these parameters, and then these parameters would impact the responses that we see. So we can go back and do that with the model. And so the points here on both of these curves are just ways to characterize sort of how steep the slope goes up with an effective diffusion constant and what the time delay is before we see the second signal. And you can see the variation in the experiments. That's the black points and the gray bars around that. And then all of the colored lines are taking one set of best fit parameters and then varying them as if the wound changed size around that. Make it either smaller or make it larger and see how it would vary the response. And we get curves that vary in the same sorts of ways that we see in the experiment. So that was a nice confirmation that this was on the right track. The other one I told you about was that the modeling suggested, we tried to fit it, that the protease source actually had to be slow. It wasn't instantaneous. And so we treated that as a prediction, went back to our experiments and looked at what was going on in the cells. And when we looked carefully at the nuclei, what we saw is that, yeah, there were some cells that were lost immediately upon wounding, but then there were multiple rows of cells back from that that disappeared over the next 90 seconds. So there was this slowed lysis of those cells. They were damaged severely enough that they were going to die, but they weren't immediately lysed open with the wounded cell. All right. Now, just to close this up, the next stage we'd like to do is to expand this model to include the intracellular calcium dynamics. There's a lot we haven't explained, right? All that interesting flaring, which to me is the most interesting part when I watch those movies, we haven't explained it all yet. Right? And so we can put all of that in, right? You've seen all these sorts of cartoons in biology papers that describe mechanisms and signaling pathways. But as systems biologists, you also realize that every one of those arrows in one of these cartoons is a differential equation, right? And so we can actually put all of these together in a model where there's a sheet of about 4,000 cells, and inside every one of those cells are all the differential equations for calcium signaling. And then those are coupled to one another with gap junctions, letting small molecules diffuse between cells, and that overarching expansion of the ligand and protease outside the cells. And if we do that, this is a first pass at it. My student Aaron Stevens really drove this. He's been amazing in making this model work. We're going to have some cells here in black that are completely ablated. These pink cells are going to then die later over time, and these green cells are undamaged by the wound itself. And if we do that, this will fade and show you the calcium signal. We get a little bit of its first expansion through the gap junctions, and then something that qualitatively, Aaron and I are both perfectionists, and we're not happy with where we are right now. But there's a lot to like about this, but it's not completely right yet, right? We do definitely get these different classes, right? Some cells where calcium goes high and stays high, others where we get the spiking and the flaring behavior, and some that just see a couple of flares, right? That's really nice. But obviously the spatial temporal characteristics of those flares don't match up exactly to what we had before. So he's revisited the model and tried to tweak parameters, and I think we're pretty close to it now in terms of having a good match there. One of the other nice things that it does is we can reproduce the knockdowns with the model. For example, here's the knockdown of the gap junctions. You get the speckling on the left-hand side. Once you lose the coordination, you have the flares on the right-hand side. Or we can do the G alpha Q and jump the gap experiment. And it'll trigger it on the other side, right? So I think at this side, I'm happy with the model structure. I really like the fact we can do these in silico knockouts and they work correctly. We're sort of in parameter tweaking and making sure we've included everything that we need to manage the experiment. But with that, I will thank the folks who did the work here. I mentioned James O'Connor already from Andrea's lab. He's been the workhorse on the experiments, helped by Bushra Akbar, who's an undergraduate who did an amazing job with all those wing disc experiments. James White is another graduate student who I showed you some of his images early in the talk. He did a lot with the syncytia and the cell cycle stuff, so we're following up that. And then Aaron Stevens is the graduate student in my lab who's been leading the modeling effort. And with that, I'll thank you and take questions. Yes.
I know you mentioned like the size of the last time you looked at it, like different like ovals and there's some changes in that because obviously like women don't always see it as simple.
That's okay. Great question. And we have that capability to do it. It's sort of there's been too much on our plate. We haven't gone down that path yet. But yes, I'm very interested in that because one of the interesting things that happens is, for example, you make a line wound. So you end up sort of with this elliptical response. There's interesting ways in which the curvature of the front as things propagate out can be important. And so you might expect different responses from sort of the broad sides as opposed to the narrow sides of the ellipse. But yeah, we're looking at that. We could do multiple wounds and see what happens with that. One of the most interesting things that we've got going on right now is that we've been desperately trying to find a way to get rid of the first expansion, right? What if we got rid of the calcium signaling completely? What would that do? There's no genetic way to get rid of that first one because it's just a physical process, but we can change how we make the wounds. So in our original ones, we were making the wounds with a single pulse, right? Because we wanted to look at the dynamics. That's what we were really interested in. If we can give that up and say, all right, I'm willing to give up the time resolution and let's make the wound in a slightly more gentle way. We can turn the laser power down and instead of hitting it once, hit it a whole bunch of times, 100 times or something like that, and raster scan over the area we'd like to make the wound. Actually, we had an undergraduate from Georgia Tech with us this summer who designed the microscope, programmed it to do all of that, works beautifully. And what we don't see that first influx of calcium, and we lose the syncytia formation. That's the first thing we've noticed. So that syncytia formation seems to be linked to that first influx of calcium. It's sort of one string. Now we're going to pull it, let's see where that goes. But yeah, playing around with how we make the wounds can lead to some very interesting results. So you have another one. You have one. Go ahead.
Yeah, I was just wondering, have you checked the efficiency of the protease? Because I think maybe there are very different types of it. Very kind of
That's a great question. Oh, so the question for those online, have we checked the identity of the protease or investigated the identity of the protease? I have to say that was one of the big fears when we wrote the paper, that the reviewers were going to ask us to do that, because that's a really hard task. We have not yet. And the reason we have not been able to do that is that you can do sort of informatics on the GBP sequence, and it's got cleavage sites for serine proteases, for cysteine proteases, for metalloproteases. They're all like within one or two bases of one another. So it very well could be this general mechanism, whatever kind of protease is released, this molecule will respond, will be cleaved by them. We certainly are. Trypsin is a serine protease, and clostropane, I don't remember what kind of clostropane is, but there are two different classes, the two that I showed you, right? And those both activate it. So we haven't been able to narrow it down and say exactly which protease it is. It may not be one. It may be a whole bunch of them. Yeah.
Yeah, so obviously right now you're doing some fruit flies, but how long do you think it will take, or how confident you think it will be to actually start doing something? Because I've had this conversation with nurses before, they talk about how these semis to get better like wound care. Obviously, because like it takes so long for something to heal. So do you think this would be applicable in like a hospital setting?
Well, so we're doing sort of very fundamental work on it, right? So it's always a long road to that. One of the things we are talking about with our next renewal of the grant that we're putting in is, are there mouse collaborators? That would be the next step to go to, right? Finding someone who's expert in mouse biology that we can extend these experiments into a mammalian system. And that's where we'd have to go. And then it's years from there still to humans. Yeah.
The precursor work that like set up the experiment on like an in vivo system, like to do it directly on a pupa. What did that, like, what was that? vitro experiment or in silico experiment, like where did the idea to do like the pupa and wound and like where did that come from?
So the question was, where did the idea for doing this in vivo come from? What were the in vitro experiments that led to it? I have to say actually that there weren't many in vitro experiments that led to it. My group had been making laser wounds and fruit flies for 20 years almost, I think, since I was a postdoc. But we were doing it for a very different reason at that point. We were interested in the mechanics of tissues. And if you make really small wounds, you can look at the relaxation and use that to figure out what's going on in the mechanics. This was just a nice extension. Andrea's lab was very interested in wound healing and basement membrane. And it was just this, the two coming together and saying, oh, we could do this. Let's see what we see.
How deep does a laser propagate into the cell line?
Good question. How deep does the laser propagate into the cells? This is a surface epithelium, and the laser, when it makes the plasma, right, we're using a very high numerical aperture lens, so it's very tightly focused.
Okay.
There is laser light that penetrates more deeply, like,
Damage doesn't.
but damage doesn't, right? Because damage is very intensity dependent, and so we only see that right at the focus.
The reason why I asked like those leading up questions, but when you do like an explant or make like a dermal cell line within a cell plate, like have you thought about conducting the same experiment? Are you concerned that that intercell protein that is cleaved by those proteases doesn't exist on a cell line? Or I'm just wondering why well, obviously like this work is working in vivo systems, but like to kind of piggyback off of Heather's question and how can we like apply this to human systems, like the in vitro experiments with human systems could be like very simple if it's a single cell line and the laser only propagates like only as deeply as one like cell length. You know what I mean? Am I making sense?
Yeah. So the question
Is
is,
that possible?
the question is essentially complications from trying this on cell lines, right? As move towards human studies or explants. Explants I'd be a little bit more confident in,
Okay.
right? Because it's clear there are some systemic effects, right? We have these GBPs that are created elsewhere and transported to the tissues of interest. So an explant should bring anything like that along.
Are they created elsewhere?
In our case, they are. The GBPs are made by the equivalent of the fly liver,
Oh,
what's called the fat body, and then they're in the circulation.
okay.
Yeah. And so what I would worry about with a cell line is that we would miss those systemic effects, right? We wouldn't have, if these sorts of peptides are present and this sort of mechanism works, that the cell line itself might not make them.
But you can't artificially add them to cell media like you did with the lysis. Like
You
the
could,
lysis.
Okay. But we don't know, we don't know the exact ones for humans, and so we wouldn't know what to add yet, right?
Right.
Yeah, I will say that this systemic effects, one of the interesting things about it is that it ends up then coupling this wound healing response. Nutrition of the organism has a big effect on how much GBP is circulating in any one time, and aging, right? So there's this interesting connection between those three that we don't clearly understand yet, but there are hints that these three are connected to one another. Yeah.
So you showed that mechanistic model in 3 slides before where you had these where you showed that the cells were getting damaged and like the proteases were coming out and then the signal was transmitted, right?
Yep.
But that model was not able to explain the first burst and the other things, right? We were only able to explain the second expansion through that model, right?
That's right.
So that, what do you think is the gap in our knowledge in terms of that mechanistic model that it is not able to like emulate the exact first burst and stuff?
So the question was, the model we showed with the ligand and receptor and the proteases only explained the second expansion and not the first. So what are we missing? I think actually in this case, we're missing stuff that we understood pretty well. And so we just ignored that part of the expansion, right? The first part of it really is just this collateral damage, making pores in the cells and calcium rushing in. We could have added that, right? And it would just sort of been this part on top of it, but we would just focus on the part that we knew was related to the proteases and the ligands. Yeah. Yeah, so we've put that in, right, in the larger model where we put the calcium signal, we put all of it in, whole kitchen sink.
All right, let's thank the speaker for the presentation. We'll close that down, and then we'll open it up to the follow-on. So
Okay.
thank you all for attending, and we will see you next time.